So, without further ado, I'd like to introduce our keynote and conference kickoff speaker, Dr. Francois Lamontagne, who will be speaking on the subject of integrating research into clinical practice, the challenges and solutions for Canada. Dr. Lamontagne holds a master's degree in pharmacology and another in health research methodology. In 2020, he launched the Canadian Clinical Research Network to improve clinical research capacity and coordination across Canada by collecting and reporting information on the conduct of clinical research within the Canadian health research ecosystem. As we were doing research for this conference, we came across a paper entitled Integrating Research into Clinical Practice, Challenges and Solutions for Canada, written by Dr. Lamontagne, his colleague Catherine Rowan, and Gordon Guyette. We could think of no better person to introduce the theme of this conference. The paper discussed the limitations of the existing clinical research infrastructure in Canada, also describing the mechanisms implemented to successively embed clinical research into the UK health system. The paper went on to further identify the traps that Canadian stakeholders must avoid, thus providing a roadmap to a Canadian version of the UK model. We're excited to introduce Dr. Lamontagne and his work to you.
Well, thank you very much, Anne-Marie, and for this introduction, and Barry and all the organizers for this meeting. It's an honor, and I'm quite excited to be here, and look forward to the discussions we'll have over today and tomorrow. So, and thanks for this very kind introduction. As was said, I was invited to present the content of this article that we published early during the pandemic. And rather than just transfer into slides what was on the paper, I thought I would give you a little bit of context and explain what led to this paper being written in mid-2020 and what we've done since, rather than maybe talk about a model that was very inspiring to us. You mentioned City. We, my co-authors and I, were quite inspired by the NIHR model, and I hope Dr. Corey will appreciate what I have to say about the NIHR, but perhaps explain what we think we can do and what we've started doing to perhaps get there differently in Canada. So a little bit of background. I did med school here in Montreal, and then I trained as a general internist and as a pharmacologist before changing direction and moving to Hamilton, McMaster, where I trained as a clinician scientist and as a critical care physician. So by the time, I'm a slow learner, by the time I was eventually recruited in Sherbrooke, I had been funded by CIHR to launch a clinical research program on resuscitation strategies in the ICU, right? So, and I started doing that, and around the same years, I was also increasingly involved in guidelines work, perhaps owed to the fact that I was working with people from McMaster. And so I fairly quickly developed a perspective on research that was multifocal. As a clinician working in the ICU who consumed a lot of research, as a person making guidelines who needs to access a certain type of information to create trustworthy guidelines, and as a researcher very much wanting to initiate and lead some of those studies. And from whatever perspective you were, or I was looking at research, the bottom line for me was that it's really hard. And it's hard. It's not hard. I mean, it's hard to train. It's hard to get funded. But I mean, beyond that, once you're quote unquote successful and you've been funded to deliver a study, the deployment and delivery of a study within a health system is difficult. And I'm not whining here as a researcher saying, it's really fun work. But the reason it's hard to deliver studies is that it's hard for non-researchers, provided they're interested and they want to collaborate or support the research endeavor. It's really hard for other stakeholders, clinicians, patients, to sign up and contribute. Our system isn't, there's no, sign up button, and that will automatically allow you to contribute in many, many places. If you're a clinician and you want to support a clinical trial, as a colleague from Sherbrooke might have said recently, you kind of have to set things up yourself. And this sort of explains why it's so hard to finish a study. When I was at McMaster, I was fortunate enough to work with methodologists and contributed to a number of methods papers, including this one, where we surveyed 6 REBs across Canada, Germany, Switzerland, and surveyed the clinical trials approved by those REBs over a period of 3 years. And it turns out that it wasn't just my own sort of personal impression. Finishing studies is difficult, and a quarter of all those RCTs that were successful, right? They had been funded, they had been reviewed and approved, and they were launched. Not half, but a quarter of those RCTs were discontinued. That's a like sizable proportion. And the main reason for discontinuation was that enrollment was poor, right? Too slow, or so it wasn't just me. It seems like it's a pretty common theme. And like some of you could say, well, suck it up, right? That's the life you chose, and that's fine. But the thing is, that has a real impact on the type of research that we get, whether we're a person with lived experience or a clinician or a guideline maker. It impacts research quality. So you have to understand that because it's so hard to finish, it creates a very, very strong internal incentive to design studies that are feasible rather than the studies that we need, right? And there's a vast amount of literature about this. Smart people, these are colleagues mostly from the US, who have, studied and written about the multiplication of studies that are too small and underpowered. And we know that that's a problem, and you can't fix that with meta-analyses necessarily, right? And they've gone as far as calling that problem unethical. There are other problems, other than size. It's very, it's quite a bit more interesting and feasible to enroll close to home, where it's easy to recruit patients, but that leaves huge parts of the population or regions not very well represented by the studies that we conduct, and that sort of fuels health inequities. And there are all sorts of other problems, right? We sometimes pick the wrong outcomes because they're easier to measure, or the wrong interventions because they're easier to measure. So this definitely, this broken ecosystem sort of affects research quality. And because of that, not that we're not productive, there's a large volume of research knowledge being created, and it's increasing, and it's perhaps excessive. It just doesn't fix the scientific uncertainty. So if what you are getting are underpowered or flawed studies, a lot of the tools in our toolbox, the healthcare interventions that we deliver, are of uncertain, the evidence is uncertain, and therefore the effect of these interventions remains uncertain. And again, there's a very vast amount of literature about this. There's empirical evidence for this. This is one recent article published last year by Jeremy Howick, incidentally, is a Canadian researcher working in the UK, and they picked a random sample of 2,500 health interventions in the Cochrane database, and they objectively appraised the scientific certainty for each of these interventions. And it turns out that a tiny proportion, so just for about 12% of those interventions, the body of scientific knowledge in support of those interventions was high. So it's actually a rare event that you have high certainty that you know what an intervention will do. And it's actually worse than that, because sometimes you've got high quality evidence that an intervention doesn't work. So if you focus on the beneficial stuff, the number shrinks to about 5% in this analysis. So, we don't necessarily get the research we need. There's high uncertainty for most of the things we do in healthcare, and it's a problem. That ultimately leads to either overuse and or underuse of interventions, overuse of things we should not be using, or underuse of things that we should be implementing rather than studying. And that's a pretty pervasive and important issue, right? Because at every time we make a clinical decision to investigate with one test or another, or to treat or not to treat, all of these are crossroads. They're very, very binary decision, but if you choose not to do something, it's an active decision to not deliver care. And unless you have high quality evidence to support this decision making, there's a very high likelihood that you'll pick the wrong direction at one of those crossroads. And so to me, that it's a pretty big deal. We realize that this is a big deal sometimes, but when we realize it, we see the tip of the iceberg, and I'll explain why. When we become most aware of this is when there's a landmark publication about a medical reversal. I mean, that's one situation where we'll come aware of this. A medical reversal is when you study a health intervention and conclusively find out that what you've been doing for a while with something that was available and administered or delivered across health systems was actually ineffective or harmful. And so when I speak to, like, in the area that I know, in that little niche area of acute or critical care, there's a endless list of such examples. So antiarrhythmics after myocardial infarction, aggressive blood transfusion therapy, which is a landmark Canadian contribution, and you'll recognize the name of John Marshall here, who contributed to this landmark work significantly. We give too much antibiotics for too long, which has economic, and antibiotic resistance implications. Forget just the direct complications for patients. Too much renal replacement therapy, beta blockers perioperatively, which ended up being harmful. So the list goes on and on and on. And it's not much work. Like, I know nothing about oncology, really, or not much, but it's not much work in any area to find similar examples of medical reversals, right? So things that were readily available and done regularly over many, many years, and turned out it was ineffective or harmful. And this is just one example. I think it was the first hit on my Google Scholar search. So they picked 3 journals, not 10, not 20, just 3 journals, a period of 9 years, and found 64 such examples of things that we were doing sort of routinely as clinicians that we ended up stopping. So that uncertainty, at those crossroads makes us do something we shouldn't be doing or not do stuff that we should be doing. With more time, we could go into the mechanisms, of why overuse is harmful. It's not very hard to understand. It's either harmful directly to do something you shouldn't be doing to a patient, but even if it's not causing direct physical harm, at the very least, there's the opportunity cost of spending scarce resources that should be allocated elsewhere. And I know that, and you could say the same for like the other side of the mirror is the underuse, right? There are a number of things that we should be implementing, perhaps. I know Barry's interested in the time it takes between approval to delivery of interventions. But even before that, there are probably efficiencies that could be gained if we realize more quickly that there's actually high certainty that something is effective. And we tend, in certain situations, to not recognize that very quickly and to continue to study stuff that's, rather proven to work. The landmark historical example is thrombolytic therapy and myocardial infarction. But there are other examples, probably particularly so in your area, in oncology, with all we've learned around precision medicine. Like, there could be perhaps accelerated processes to recognize that some things are ready for implementation. And, in a different setting, I might have a lot to tell you about how slow implementation is sometimes driven by other factors, so the willingness and ability to pay for healthcare, which is in turn a huge problem for health inequities in Africa. So this is work we did in West Africa during various Ebola outbreaks, but even within Canada, this is a significant problem. So by now you should be asking me what the point is and when I'll get to that paper. So the point is, if the research ecosystem as a whole is flawed, I think that masks the fact that we're dealing with so much scientific uncertainty in medicine, because we certainly are producing a lot of papers. They're just not giving us the answers we need or the information we need. And it perpetuates, in a way, uncertainty, because this dishes out a lot of knowledge that can lead to spurious conclusion. So scientific uncertainty, in turn, causes underuse or overuse of healthcare interventions. And this, causes harm directly and threatens the sustainability of health systems because of all the waste, limited resources. But this is something that we can fix. This is something I believed a long time ago, and this is the reason we wrote this paper that was mentioned earlier on. So this is an op-ed piece. We were already convinced that this is something that could be fixed, and then the pandemic was declared, and globally, everyone was acutely aware of how badly we needed to get our act together and produce evidence very rapidly, because everything had to be learned about COVID, and everyone's eyes turned to the UK, or because that's where a lot of the clinical evidence for the interventions was coming from. But not everyone understood how it came about that the UK was able to be so productive during the pandemic, and we thought that we needed to explain this in the paper. And the reason I signed the paper with my colleague from the UK, Kathy Rowan and Gordon, is that what I didn't tell you is upon being recruited in Sherbrooke for this trial about resuscitation strategies, we did complete this pilot RCT across 12 sites in Canada and the US. It was a success, as you would, we learned a lot about the study. We improved the design of what was going to be the next big trial that was going to be much larger and designed to inform practices, and we published it. But one of the things that we learned was that this next larger, conclusive trial was not going to be feasible here, for all sorts of reasons. And around the same period, I had an opportunity to sort of pick up my bags and move to the UK and bring with me the exact same protocol that was initially designed for and intended for Canada, and pitch it to NIHR, where it was a slam dunk. It was immediately funded, and after securing funding, we immediately were able to activate 65 sites across England, Wales, and Northern Ireland. So you understand that 65 sites means that we activated the study not in the well-known university centers around the Thames. It was activated across the board, across the entire health system, in community hospitals that actually treat patients. So this spirit that you want to conduct the research where patients are treated, that was possible there. And it was possible to recruit twice as many patients as we had intended initially with the same money within the same time frame, because we recruited over weekends as much as during weekdays, during the night as much as during the day, which it's not something you would do maybe with someone who lives at home and suffers from cancer. But if you're going to run a critical care trial, these patients don't show up between 9 to 5, and you have to be able to do this. So anyway, a successful study, but for me it was an awakening. For me, I sort of realized that in terms of research from the systems level, I was sort of it was a tale of two cities, to quote books I've never read. I'm French, I don't have to read everything. So this was my personal experience long before the pandemic, and I had that sort of entrenched in my brain that this was something we could learn from in Canada. But then the pandemic occurred, and by then I was the methods chair for many of the WHO guidelines on therapies for COVID. So I chaired the first guideline on steroids. And I guarantee you that on this first occasion and so many other occasions, more than 90% of all the data we were looking at was generated in the UK. So certainly recovery had a lot to do with it, but even REMAP-CAP, as John will tell you, the tocilizumab data that came out of REMAP-CAP, and then ultimately also from recovery, the tocilizumab data from REMAP-CAP was also generated in the UK. So this was like confirmation that the system was not only highly efficient, it was nimble. It could be repurposed and address health emergencies when it needed to. So in terms of the paper we wrote, this is it. Like the main points are those 7 bullets, right? We just thought it was important for people. Everyone wanted to gain a system, an ecosystem that allowed this in their country, in their area, but I think failed to understand what the steps that had to be taken to get there, which aren't, sort of trivial. So we explained that this was like this NIHR, NHS system was born out of political will. It was very much a top-down implementation, unless it wasn't sort of a consensus-driven process. It wasn't born out of the scientific community. It was born out of political will. And although there's a date, right? Dame Sally Davies, 2006, but there was a lot of work done before that. After, and realizing that perhaps the UK, which was the trailblazer, and particularly in cancer research, was perhaps falling behind, and there was an effort, lots of deep analyses of what was happening, and an effort to sort of improve on things, and that led to the creation of the NIHR. Now, how it happened, and someone asked me exactly how might we integrate, practically speaking, so what happened there. And you could see why there might not have been consensus and why they needed the top-down approach. They repossessed and repurposed funds that were not research funds, but that were funds of the health system. So monies ring-fenced for the trusts or hospitals that was dedicated to research, but perhaps not used the way, optimally. So they took those away, which can't have been a popular decision, giving it back under certain conditions, one of those being that the trusts had to set in place locally the infrastructure, the team, the people able to deploy the studies prioritized by the committees, and I'll get back to that, or by the specialty area on the portfolio. So CEOs, clinicians, individuals and groups and stakeholders who may not have been fully engaged with the research endeavor, all of a sudden were very much incentivized to become interested and to set things up because all of a sudden there was leverage. Having essentially nationalized the research infrastructure or ecosystem, it became very important because this was costly that it be managed carefully. So this is going to be a rare wealth, but you don't want it to go to waste. And so they had to make sure that there was not going to be too much overlap or dilution of their efforts. So managing the ecosystem became a big part of what they're doing. So in every area, a committee, is involved in setting priorities, but making sure that once a study begins, that it won't be in competition with too many similar studies recruiting the same participants. Otherwise, you dilute your effect, you slow down the process of generating knowledge, and you end up with too many small studies. So they managed it very carefully. It also gave a lot of stakeholder groups an influence in setting research priorities that they didn't have before. So before that, like in many places, research was chosen by research funders, speaking to research networks and researchers, but all of a sudden, if having nationalized this research ecosystem, which is in essence internal to the health system itself, the health system was able to start saying, there are these knowledge gaps that are not interesting to you guys, that you need to do something for us, right? We've set this up so that you can use it, but you need to do something in return. And I think that's how I benefited from the NIHR model. Vasopressors, which is what I'm studying, is not sexy. It's like the oldest drug, it's generic, no one can make money out of it. And the most that you, like positive or negative, the study ends up being how we can adjust something we've already been doing. The media aren't interested in a headline that says, oh, doctors have been using this since the 19th century and now they're using it a bit differently, right? That's not, so in the UK they call those market failures. They're market failures, but they're important for the system. They're important for patient safety, right? So we need to dedicate some of our time and resources to that type of research, and those type of research questions will be commissioned by the health system. And so there are detractors, and people who don't like the NIHR will say that by centralizing the research prioritization, they're killing initiative, they're killing, imagination. And so I think they would say, I don't want to put words in their mouth, but they would say that that's not accurate, that it's not an all or nothing deal. It's basically a careful balancing act, and an effort to balance the push and pull. So there's an idea that they can pull some research programs from the research community when they need to, but they're also encouraging smart investigators with innovative ideas to push their ideas to the NIHR as well. Anyway, and there's more to say, but I'll just move forward. I don't know how I'm doing for time. I don't have too much more to say. So in that paper, we described how it worked, and we weren't naive. Like, we knew that it couldn't be copied or emulated as is in Canada, so we discussed what the barriers were, and there are many, and we tried to explain how we might overcome these barriers in Canada with, moderate success, I'd say. But some of these barriers are, well, first off, like, there's not the same political will here, clearly. We're not there. This isn't happening top-down anytime soon. And if there were political will, health in Canada is a provincial jurisdiction, so you don't need to convince one, health minister or deputy health minister. You need to convince them all, and good luck with that. And then, assuming you convince one province or two, there's a fair amount of territorial protectionism. So for this to work, every province would have to agree to share, because the single province is too small to deliver these large, pragmatic, efficient trials, and good luck with that as well. We've got different legal systems, which bears into the laws that regulate research and the ethics of it. And there's a very strong tradition of institutional independence and philanthropy. So it's going to be a challenge. So that paper was an effort to tell my prime minister and the premier, what they should be doing. That worked well. And I realized fairly quickly that I'm not a lobbyist. I'm not good at this. And the paper was read, and some people liked it, and some people didn't. But I don't think it was read that much. And there was a bit of a pause and time to reflect. And we with my mentors and Kathy and Gordon and other people who've been involved and supportive, and Andres Lopakis is one that's been an inspiration for this, and Deborah Cook. And they've inspired us to maybe not so much focus on how it happened in the UK and how they're doing it, but what the essential ingredient was. And that's where we came to the conclusion that the end game isn't so much, the top-down, whatever, forcing people to buy into a new system. The end game was that ultimately, in this model, it was all about the elephants that were not in the room previously. So everyone that is essential, whose contribution is essential for research to work really well and efficiently, who are not currently engaged, and who are unaware that they should be engaged. They're unaware that all of this scientific uncertainty matters for them. So the patients, the public, right, who are not yet patients but who may become patients, the clinicians, the industry, obviously, and guideline makers, the public health. And so how do you get these stakeholders in the room where it happens to talk about research, to care about research, and start, prioritizing it, planning it, designing it, and delivering it, and then using it. If you're not going to use a top-down approach, well, we figured maybe one way is to collect information and share information. So all of these stakeholder groups might be aware of research in, some way. They might not be seeing the entire picture. And so our MO moving forward was maybe we can collect information and make it available to them so that they see the elephant, and then they decide on their own whether there's a problem to fix and how to fix it. So ideally, to get there, we think that they need to know, all of these stakeholder groups need to have a pretty clear understanding of what the burden of disease is in the country, in Canada, say. So cancer, stroke, mental health, and see the entire picture. Very important that they know what usual care consists of, because there's a huge difference between what we think we do and what we actually do, and how usual care is very heterogeneous geographically, even in Canada. And then you would need all those experts who are quite good at ascertaining the scientific quality underlying each of those components of usual care to tell us where the knowledge gaps are. And once you've got that, you've got a pretty good map that is dynamic of the knowledge gaps, and you can start to have discussions about research priorities. But there's an important missing piece, because you cannot appreciate whether your research ecosystem is giving you what you need unless you also have a fourth wheel to the wagon, which is an ability to track the research that is occurring within your ecosystem and whether you're getting from it what you need. So that's how we set up the CCRN, as a bit of a salute to our UK colleagues. The name may change, but what we're doing essentially is we've recruited very experienced research managers, not people who recruit patients locally in hospitals, but people who are used to managing and coordinating multi-center studies. And what they approach researchers, coordinating centers, and they collect information about studies that are being rolled out in our Canadian system, and they collect information about recruitment in real time. And that's important for us because we want to be able to track research progress in real time. And in parallel, they're also collecting a lot of information from the sites that are participating to those studies, incrementally, gradually, iteratively improving our understanding of the Canadian research infrastructure, because there is no such thing as a research infrastructure. The infrastructure is whatever the one local investigator set up at that site, right? And so we are learning about the diversity of the research infrastructure across Canada. Now, the reason we're doing this is not so much to publish papers. We want this analyzed and available on a dashboard that would be freely available to all of those stakeholder groups, and we're actually preparing reports that we're pushing to stakeholder groups, increasingly tailored reports. But we're not going to shy away from having one or two publications out of it, if it at least to exemplify what we can get out of this. And this is a quick of a peek into a paper that's going to be coming out in the near future, and not because warning Megan Bettel that I wasn't picking on CIHR in particular, but it turns out that the first data we got was for COVID-related studies. We started doing this in 2020, so we decided that the first analysis would be an analysis of the CIHR-funded studies for COVID-19. Not all of them, but the studies that prospectively recruited patients treated in acute care hospitals during the pandemic. So that's our denominator, prospective studies of patients treated in acute care hospitals. So sorry that my picture is hiding, yeah, I think the slide looks better, thanks. So there were 26 such CIHR-funded studies recruiting patients in acute care hospitals. We have data on 23. On this slide, you see 22 studies. We've been updating this since I created the slides. So 16 RCTs were funded. This is a 2-year period that we're looking at to recruit patients with COVID in acute care hospitals, 16. That's CIHR. That's not it's not FRQS, it's not the private foundations out of the Montreal Heart Institute, that's just the CIHR funded studies. They recruited together 3,860 participants from 78 hospitals. And 31, the median is 31 per site over more than 2 years. And we have the same information for observational studies. Now, by virtue of collecting information about the sites, right, that recruited, where those participants were recruited, we can tell you if you pool the sites that contributed to RCTs and or observational studies, the number goes up to 97. 68% of those research active sites had access to a research pharmacy, so 2/3. That's pretty important if you're going to run a drug trial. We know that those sites activated the median of 2 studies at every site, but some sites activated far more studies. All of these were recruiting COVID patients, so that means sometimes they had to make a choice, right? The same participant couldn't be recruited to 11 studies, so they probably made a choice. They employed a median of 1.3 research staff. A third were able to enroll outside working hours, and about a half were familiar with deferred consent models, which were sometimes important and a rate-limiting element. So by virtue of having data that are study-level characteristics and site-level characteristics, we're able to build this, and this isn't me, other smarter people are able to create a fancy multivariable, multi-level model to measure these associations between characteristics of either the studies or the sites and ability to recruit efficiently. And these aren't the final, the reason I'm asking not to take pictures is you can take the pictures if you want, but that's not the final data, it's just, consider it mock data. But we learn about variation, by province, by study design, by research infrastructure characteristics, and we learn about the unexplained variability in recruitment adjusted for all of these things. There will always be differences in how easy it is to recruit in different studies. Some studies are just easier or harder than others. So, if you just give a pill and count bodies after 20 days, that's a simple design. But if your intervention is more complex and the follow-up is complex and you're drawing blood, that will slow recruitment. It will always be that way. But once you've adjusted for everything else, there shouldn't be that much variation across sites. Now, that's something we can continue to understand better and maybe try to improve upon. You can map using this, you can map out where recruitment is occurring. So the red dots are those 97 research active sites. They're drowned in a sea of blue dots, which represent the research inactive sites. Well, there's certainly work to do here in Canada to build up our research capacity. And not surprisingly, because of this, a lot of our champions in clinical research in Canada, they're champions because they recruit outside Canada, which is great, but perhaps, we need to fix a few things here as well. I'll skip over this, but it's a tool that's used not so much to publish, as I said, but to manage our research ecosystem. So because we have recruitment in real time, funders, network leaders can actually follow the success of or the progress of a research, of a study, before the end of grant report. And maybe, as they do in the UK, pull the plug on a study that is failing because those scarce resources should perhaps be used differently. This is an add-on project led by Nicole Yada, who's in the room here today, a health data expert, and Denis Boutin, a patient partner. You would think that research should be occurring where there is disease activity, but in fact, research is happening where there is infrastructure and interest, which is a bit of a problem. So what they are doing is mapping onto those maps that I showed you, another heat map of CHI data showing COVID hospitalization. And again, this isn't the final data, but you can quickly get a sense that there is a bit of a discrepancy between disease burden and research activity. Now, if you're trying to convince people to fund more RCTs, we believe that by carefully monitoring research is one way to do it. This isn't something we can put in a nice map or slide, but on the website there's a wealth of data characterizing the data that is collected across all of those clinical studies and the specimens that have been collected for those studies. Now, you have to know that during the pandemic, I didn't show you this because that wasn't funded by CIHR, but there was a COVID biobank funded for COVID, which was, and I'm an ally, I contributed, I'm part of I think it was important, but it was funded at the cost of I think we're close to 20 million, to create this big COVID biobank. Interestingly, and I know this because I sit on the board, the biggest cost wasn't the DNA, RNA, proteomic analyses. The biggest cost was paying clinical research staff to screen patients, get consent, collect blood, collect data. All things that were already being paid for by all of those funded studies. So wouldn't it be nice if all of the clinician scientists who run cohort studies or trials had preemptively gotten consent and offered patients, an opportunity to contribute to this national biobank, which would evolve on the back of a growing research portfolio? All of the money spent for that standalone biobank could be used differently, and clinician scientists could kill two birds with one stone and answer a clinical question and populate this massive biobank. All that it requires is a little bit of foresight to embed in the consent forms permission to do this and documentation so that we know exactly what data exists in what data set, what specimens exist and where they are. And so we're, for now, just we're the Tinder of researchers and just putting on a platform the information and people can decide to hook up on their own. But at some point, maybe we want to give a thought to whether this should not be mandatory. It would certainly be an efficient model. And so a better return on the investment is perhaps a way to better fund trials. In the UK, they mandate economic analyses for every trial, and maybe that's one way to get health ministers or deputy ministers of health to care about research. If they know there are cost savings at the end of it, sometimes they might feel they have a stake in better clinical research as well. And I'll skip over the last few parts, but this is something you can use for benchmarking, comparing provincial health systems, comparing sites anonymously, at least figuring out which sites are the top enrollers and maybe, creating that warm base ready for the next crisis, those sites that you can go to, but also learning from the top sites so that you can copy their recipe at sites that, maybe have to improve a little bit. So, long story, I'm done. I told you why I became interested. I told you what I witnessed. I told you about this firsthand experience of a very different and inspiring model. And I told you how with that paper we were telling people what we thought we should be doing in Canada. That doesn't work. We're researchers. What we do is measure things. So what we're focusing on now is understanding how the research ecosystem works, with a view of implicating more stakeholders, patients, clinicians, administrators in those discussions so that perhaps now immediately we can better use the scarce resources we have more efficiently, but at some point enhancing our research capacity and create this learning health system that everyone talks about. That's it for me.
Have questions, please feel free to come up to one of the microphones, or if you've got comments as well. Right. Jerry's being very gracious and allowing Linda to go first.
Thank you very much. That was excellent. Thank you so much. Linda Hunter, CIHR CanTrain, one of the clinical trial training programs that's been funded, and a patient representative. I'm wondering kind of three interrelated things. It sounds what you have created is something that might be, I don't know, I'll use the word a framework or a model or a gold standard of how to put something together for others to learn from. I'm wondering if that's a thought in people's mind of doing something like that. You mentioned patient involvement in one of the studies that you discussed. However, I'm wondering how much patients have been involved throughout the process of looking at this situation, let's call it, in Canada. And then I'm wondering, you talked about clinical research professionals, very diverse across Canada and very different in maybe what they do. Is there a need to have enhanced or better or changed strategic direction in the training associated with those people? So kind of interrelated questions, if you don't mind answering those.
I don't mind. I might need your help to remember. I might not answer them in order. But patient partners are involved in this CRN work. So they were involved in the planning of it and the rolling out of it. And one example, and they're involved and increasingly involved. This is very much iterative, and we're always looking to learn and improve. But one concrete example of this, which actually came from Denis Boutin, the gentleman I just mentioned, who he was part of the discussion, and initially we were just focusing on choosing what data we'd collect and how we collect it, and but he said at some point, looking at what we were using and analyzing and putting in reports, he said, you're not collecting any information about whether or not and how, if they are indeed implicating or involving patients in a given study. You're not describing how patients are involved. And so he's quite not concerned. He's aware of tokenism. And so he said, well, why don't you, this case report form, or these tools that we use to collect information to describe the research portfolio and the research infrastructure at sites, we're now going back to those sites and to all those studies to ask them if in their leadership, like the steering committee of those studies, patients are involved in what it is that they're doing. And in many cases, they're not, or they're not doing much, right? And but that's just one concrete example, but we're eager to do more and better. In terms of whether it can be a framework, well, I don't know whether your question pertains to a framework for studies or but I don't, I think even in the UK, which I'm in awe of, there isn't as much effort put into steering committees in the UK don't mean the same thing they mean in Canada. Steering committees are somewhat independent groups that interact with researchers and report to the funder and ultimately decide whether a study should continue or not. So there is this process for monitoring, but there isn't a publicly available dashboard where everyone, from the public to the health administrators, can understand what the research portfolio is and track it in real time. So in that sense, yeah, it can be replicated. We're more than happy to do this for others. We've started having discussions with international research networks who quite like this, the front end of it, the website, the dashboard, because it's intended to, the website is designed so that people can pick a study or any grouping of studies and run the same analyses that will appear in that paper, so they can see recruitment. And so it is a framework. But your last question, about the research infrastructure and whether we can, it's extremely diverse, and I can't tell you, even what I showed you here, which seems very simple. Research pharmacy, simple question, right? It's a yes or no answer. Well, actually, no. It's pretty complex, because a research pharmacy in the downtown Montreal, it will be a different building with different pharmacists trained differently to do research. In some other places where they have a research pharmacy, it's actually the hospital pharmacy that allows a few pharmacists to train and do something a little bit different. But it's the clinical pharmacy that does some research work, and in some places, so anytime you ask a question about research infrastructure, and this is why we employed such experienced research project managers, they're in the business when they run a multi-center study of negotiating a protocol in places that do things very differently, but we are impressed by how much we have yet to learn just to characterize the research infrastructure across Canada. It's extremely heterogeneous.
Thank you very much. That's great.
Sorry for a long answer.
Is there another?
Yeah.
Thank you very much. Excellent talk. I think One of the most impressive things that you said, or impressed on me, was the top-down nature of the UK transformation. In Cancer Research UK, it was extremely important, and we also admire what they've done here. For example, in Quebec, we have the Ministry of Health, and then we have the Ministry of Economic Development and Innovation, and they one thinks it's administering health, and the other thinks it's investing in healthcare research, and they don't talk to each other, they don't understand each other, they don't coordinate at all. And so the concept of integrating research into healthcare is a real challenge. So on the one hand, we need a top-down thing, and I've been talking and trying to convince potential industry partners, even with government, that they need buy-in from both ministries. On the other hand, I think we need a bottom-up, not just patient involvement in the trials, but I'm starting to believe that we need an actual movement of patients, a bottom-up movement of patients for clinical research as part of clinical care. And in other words, something that will move the population, move the electorate, and therefore move the politicians. And I'm inspired by the 1980s ACT UP group in the HIV treatment development, where they just simply challenged the FDA and all the regulators.
Yeah.
So I think we need both, but and the goal is integrating all of these things. So I don't know if you want to speak to that.
Well, I mean, I absolutely agree, and it's actually the motivation, you know, behind this project is when I said, rather than telling our VIPs what they should be doing, we'll just measure what we can and make it, you know, put it out there. So that all of these groups, including the public and patients, can sort of see on their own and that they can decide whether it's important for them. I think if we do the work, I think this is so crucial that people should quickly figure out that it's important and there's something to be fixed. And I certainly hope that it can be a vehicle to pressure, you know, our decision makers to, you know, get their act together. So that's kind of why we're doing it. And I like your reference to the HIV. The challenge here is that it has, I think what we want is, because it's a single, I skipped over the network sort of figure, but it's an ecosystem, right? And much like in any ecosystem, you've got different species interacting, sometimes synergistically, sometimes they compete. I think we want to avoid doing it for one illness, one disease. And that's what we do right now, right? So you've got the diabetics, they're a strong lobby, and they are, or cancer or heart disease, and they're going at it. So we've got the stroke network in Canada that came out of an NCE, and so now we've got a great transform care for stroke. Now, if you want the system to be transformed and to be able to continue to transform, if you want a learning health system, it has to work it has to be agnostic to an illness. So you need to mobilize the public and the patients, not people with one illness, and that's the challenge. But you know, that's kind of what we're trying to, you know, contribute to with one small brick. But I totally agree.
I'm going to ask everyone to thank Dr. Lamontagne.